Ladies and gentlemen, my presentation is I put in such a way that the future food security and the role of genetics and environmental interaction, how those play into address the global food and agriculture sustainability. And this is I quickly go through the Earth, very unique. This is the only place, as far as we know, in the whole universe is livable to intelligent creatures like us. And we have a best atmosphere, is a gift. And now, last nearly after the Industrial Revolution, this being challenged. And we all is aware of this thing, and we all need to work hard to tackle this issue. The primary culprit for this issue is the warming and climate change is the CO2. Carbon dioxide concentration since the Industrial Revolution is increasing. If we see in this figure, it's 1800 years of the CO2 concentration in the atmosphere. Last 160 years is rapidly increasing. It has never been 300 parts per million during that period of time. Since 1800 that the concentration started to increase. It is now nearly 423 parts per million. It used to be below 300 parts per million. This is a big increment. And so it is always the CO2 concentration is associated with the temperature change. We expect the temperature to change, go up by 2 to 4 degrees by end of this century. And the precipitation is a temperature change is a major culprit, and it also accompanied the changes. The precipitation, it's become a more erratic and a more frequent as a drought become a more common in the future atmosphere. These parameters have a major impact on the Earth physical and biophysical processes. So therefore, it is very important for us to tackle this issue. So I want to share this one, and I thought my work related to the climate change is 1994. And we published this paper in an Australian Journal of Plant Physiology, and it's a part of my PhD. And what I said was the CO2 concentration in the atmosphere, 350 parts per million. Today I work on the CO2 space, and I write now 423 parts per million when I write today. So itself is 20% of this substrate. The CO2 is increased in the atmosphere. Again, we need to remember CO2 is just not only the greenhouse gas, it also the substrate for the photosynthesis. Whole food security photosynthesis is driven by this very special ingredient, and this is not the most of the C3 plants in the plant kingdom. We have three major biochemical types, C3 and C4. C3 is not saturated. When CO2 concentration increases, photosynthesis also increases. And so, on the other hand, we also have another issue, is the population is not settled yet. It is increasing, and right now is around 7.8 billion. It's going to be 9 billion by middle of this century. We need to feed these people. There is no other alternative. We have limited land. We need to improve the land sustainability and same time adapting to the climate change. And if you see this graph, for last nearly 70 years, the population increased by 2.6-fold. At the same time, cereal yield increased by nearly 3.1-fold. And the cereal production increased by 3.5-fold. So we can see consistent increase in the cereal production around the globe. And if you closely look at here in the right panel, right, it is now little bit settled. There is no yield increment, and so this is one major factor is a climate change, and also other factor is a biological factors that we have not maybe addressed, but in the Western world, they're working hard to get this thing up and running and get the yield increment is regularly to get it running. So this is, especially in this part of the world, this is one of the big issue. This is the gentleman is Norman Borlaug, who brought the green technology is nearly 70 years back. He introduced two things. One is he brought into the nitrogen responsive genes into rice plants, and also they introduced the stunted gene like growth into the rice system. So through which somehow he was able to increase the rice yield potential, the cereal yield potential by 3.3 times, and also population expanded from 2.2 the 5%, and the per capita the rice, whatever the cereal production was able to increase. This is how we recovered from that kind of the food security, major food security issues 70 years back. Now again we have come to the same point, similar point, because we have a great population, we have the very complicated, the global warming and climate change. And if we see today, this is a kind of the deficiency, food security issues in the world. Nearly 8.9% of the people are starving. Starving in the sense that there are issues in a different degree of food issues, nutritional issues. And among them is Africa is the worst. So nearly 20% of the population is going through the like a food security issue. And now this thing is picking up, and it's a very high time to look to address this issue as soon as possible. This kind of gathering, I think, essential, and we need to have more this kind of gathering across the world to get this message to the people and the public and also the bureaucrats to work on and invest and tackle this issue. So future going to be is a population we know is somewhere around, it will be by 2050 9.7 billion, and also is a food demand will increase at least by 50 to 70%. And the climate change is we're going to have a major extreme weathers, and the temperature going to be high, and also growing season is going to be changed. And same time, the projected yield, driven by the climate, will decrease by 10 to 25%. It's depend on the region where you're coming from. And it's a urgent, what we want to say, the urgent action need to develop a climate resilient agriculture is all over the world, across the globe. And the most important thing, I think we need to rethink about the second green revolution. This is a very high time. The second green revolution is a holistic, it's a multidisciplinary, the biological and chemical revolution. We need to, it's not first the green revolution is very much around the genetics, but this is the genetics and the environment. That genetic and environment need to optimize. I don't think we have, especially this part of the world, we have addressed the fully. For example, if you take rice as a high genetic the potential, we have not capitalized, and equally we have a beautiful climate environment that also we have not capitalized. We really need to our cropping system, we need to optimize for these two things, and to go and try to achieve the best the yield potential of the crops. I think this is a story. This is a multidisciplinary. This comes of biology and also engineering, and all other the science need to come into play to tackle this issue. So the first, as said, is a first green revolution is around the genetics, and the second green revolution need to come. It is genetic engineering, the climate resilience and precision agriculture, and also sustainability around, and digital technology, and so many AI applications, etc. I'm not going to go into this. I want to say it's a truly multidisciplinary approach for the second green revolution. And what I want to touch is the CO2. This is my career was last the 30 years I was working on the climate change and CO2 and the role that play, how important in terms of the food security. And when come to the food security and CO2, I ask six questions. The one is how elevated, because it's a substrate, it's increased. How we capitalize this substrate to improve the photosynthesis and the food security and nutrient security. So first question, how the elevated CO2, how climate change impact on the plant physiological processes. These are the fundamental information that we need to generate if we want to really address this issue. And the second is what are the implications for crop yield and quality, because you can improve the crop yield, but if crop quality is compromised, that also need to be addressed. That's been already happened. And the next question asked is the grain yield and the nutritional property, because that's also linked to the nutrition of the people and the public. And so again, we want to ask the broader question on the impact of the global food and the nutrient security. So finally, we want to ask, can we address some of the challenges in grain yield and quality under the extreme climate? This is the questions I ask all the way my career. I'll pick up a couple for this presentation. And this is something, like a lot of climate change research in the Western world is familiar. The technology we use is very known. But when it comes to the developed world, we have limited resources, and it's very difficult to do that type of experiment. These are the experiments, especially the CO2 and the and temperature type of studies need to do. Is a growth chamber studies you can do, and also they're called technological pre-air CO2 enrichment studies, and the open top chamber studies, and temperature manipulation experiment we call TIIKM experiment, and drought water stress experiment models, and the combined stress experiments, etc. So these are very high tech and also very expensive investments. And quickly, I will share a couple of experimental models that I involve with growth chamber studies. Is very much the control experiment, and the open top chamber studies, and also the field experiment, the field CO2 enrichment study. And also we can manipulate any kind of environmental conditions in this type of experiment models. The temperature, CO2, this is the two major culprit that climate change is impact around. The CO2 has a positive effect, the temperature has a negative effect. Mostly in the most part of the world, in the tropical latitude, temperature overpower the CO2 effect. This is the fundamental, the behind of the climate change. And what I've said is that this is a very expensive investment. So I involved with this experiment, and Gary is here, we two were there, from the setting up to the finishing of the experiment. This is one of the beautiful experiments. It's the Southern Hemisphere, the biggest experiment. And so we generate a hell of a lot of data. And this is the data shared in all the climate food production models around the world coming from the Southern Hemisphere. And so where you can test all the experiment variables, CO2 and the temperature, water, nitrogen in the field level. And the cost of running this experiment, maybe 1 year, you need to have $3 million US is close to just for the infrastructure and the CO2. So this is the cost involved, but it's I think for Sri Lanka it's very hard to do this type of experiment. At least we need to think about running similar small experiment and generate the data. That data is very essential for us to adapt to the future climate. And this is some of the information we generated, and the CO2 is a primary factor for the photosynthesis. And most of the CO2 plant, as said, is not photosynthetically saturated. This graph shows how photosynthesis response varied under different CO2 concentration. What I want to say is, this is with the rice, when they put into high CO2, short term, there is they do respond, but when they expose to longer period, they adjust and acclimate. this process called acclimation to the climate, especially the CO2. That initial stimulation of the photosynthesis will quickly disappear, but we cannot capitalize that initial gain of the photosynthesis. And what happened is a main driver for such change is the one of the key enzyme is the rubisco. Rubisco get readjusted to that kind of climate. Rubisco is one of the major protein on the earth. That's the largest amount of the protein, and this protein level decrease the expression of the genes. There's two assembled whole enzyme assembled by the two gene family, and so the expression of the gene also change and lead to a kind of the acclimation and reduce the amount of the rubisco. And we made the antisense construct, the reduced rubisco rise. We can see the same response under elevated CO2. Still, you can see right side figure, the black dotted line is a plant grow at elevated CO2. You can see substantial reduction. It's a very common response that drive coming from the CO2. And what we see is, what I want to say is, like these numbers maybe you're not familiar. The rubisco is the biosphere and atmosphere link. And it is a global, the CO2 emission is around 37 billion metric ton annually. And we have a rubisco on the earth is around 70 to 80 billion metric tons. This is the biggest protein, and this is the biggest protein, is the catalytic protein that put CO2 into the carbon. And at the same time, the rubisco, if we want to net zero our emission, we need to increase the amount of the rubisco on the Earth atmosphere was 14.7 billion metric ton. This is the easiest way if you want to really to go and achieve the net zero emission. And you need roughly, my calculation suggests that 1.5 trillion plants on the earth is a medium size, can do the job. And again, like I was talking to a number of organizations to start this initiative, but I will open up this discussion in the later part of my talk. And this is what exactly happened. This is what the graph shows the meta-analysis. We have taken all the published information so far, and we put together, and using a meta-analysis, we look at how photosynthesis and stomatal conductance behave. You can see the photosynthesis change, increase. There is a variation between the different biological types. See, pore plants the stomatal conductance is a decrease, but photosynthesis no change. And equally, regardless of all species, photosynthetic rates. Stomatal conductors decrease, but the photosynthetic rates vary. The C4 does not respond, but the stomatal conductance is still low. There is one message from here. Is regardless of the biochemical type of the photosynthesis, the stomatal conductors operate differently. The stomatal conductance decrease in all the species, and this is the main driver of the water cycle. So this is something, in one way is a good news. The water use efficiency of the plants increase under the future climate. And this is how, in a cellular level, it's a very complicated. I'm not going to talk about the stomatal regulation. You can see how it looks like in the right side figure. And when CO2 concentration increases, the stomata start closing down. The billion of trillions of the stomata around, and that also impact on the water use efficiency of the plants. This is something, the most important fundamental of climate change and food security. I think we all need to closely look at the temperature and CO2 interaction. So again, when the CO2 concentration is lower in the atmosphere, the photosynthetic rates are low, as I said. but this interaction is dominant. If we don't understand, if we cannot pin down, and if we cannot get this thing into our models, etc., it's very hard to predict the food production and the food security issues, because this is a fundamental information that we use for the modeling. So those parameters, I think, is a high time to take into the modeling, capture into the right way. Maybe some fine tunings are required. So what I want to put here is we put everything together, whatever our discoveries, on the basics. I always believe without basic science, it's very difficult to advance into the applications. So this need to go hand to hand. If it is 50% of the basic science, also we need to have application science is like a 50%. This is maybe a reasonable balance. All the major discoveries and all the major applications what we develop from the basic sciences. This is a classic example. What we say is this is a pathway of what we identify how it works under climate change, especially under high CO2 scenario. All cell division accelerated, and also the cell number increase, cell wall differentiation change, and the shoot apex size, shape change, the hormonal dynamics change. This all together, at the end, we are going to get into the bigger biomass. And what we found from this work is that the photosynthesis increased significantly under high CO2, and again with the temperature is a compromise, and also the increase there is a large variability in the species, and also stomatal conductance reduce, which also facilitate the water use efficiency of the plants. And this is not for the one species, this is for the universal thing. So this is the fundamental of the underneath of the climate change science, climate change biology. And when come to the field, we put this thing into the test, and we look at the agronomic, the impact. This is again done in Japanese rice phase experiment. What we grow in the plants in rice, we put into high CO2, and in the phase experimental system, and we can see rice yield increase under elevated CO2. And there is an interaction between nitrogen and CO2. When the nitrogen concentration is high, you can see the yield stimulation also high. And so as pointed out, again, this is the temperature CO2 interaction. This is something we need to get into correct in the modeling. I think still we are not in a right track. And so the CO2, when goes up, the photosynthesis increase, biomass increase, but the temperature, if you raise by 4 degrees C, you can see how it compromises the yield response. It's a heavily compromised on the yield. And there is a large genetic variability as well in response to elevated CO2. You can see this is two different scenarios. One is the right side is plants growth in the controlled system, and other one, the left side, is plants grow at the phase system, pre-AI enrichment system. And when we grow in the plant, we can see the bigger response. But in the field, this is actually percentage stimulation in this right side, but other one is absolute change. Whatever we can see is there is a large genetic variability in plant response to elevated CO2. And what we can do is, when we say there is a genetic background to respond to CO2, the CO2 there is a genetic orientation. There is a very limited work done on this one. We have seen we have mapped using a double haploid population, and we identify some QTLs that's a very response to CO2. There is an opportunity for the breeding crops in the future to take as a one factor to breed crops and to improve the crop productivity. It is a fertilizer, it is a substrate. I don't think anywhere else we have taken seriously into this consideration. That means there is a great opportunity to capitalize the CO2 in one way to for the crop breeding programs. And this is when you put all together, is a 2050 going to be the cereal production is been major impact, but when you come to the 2080, there will be a disaster as discussed, and we can see in general in 2050 to 10% like a reduction in cereal production globally is regardless of whatever the strategies we adopted. And this is I think the fundamental of the climate change and the fundamental for the food security. And I will quickly go the yield increase. There was a great yield increments and elevated CO2, and there is a large genetic variability that we can capture for the breeding programs. And also the temperature CO2 interaction is very important to capture for our modeling and to make a good right prediction of the food security is globally as well as nationally. And other area that we have ignored all the way is grain quality, and the grain quality is very important. That's all human nutrition is 70% of our nutrient coming from cereals and pulses. And what we see is CO2 has a major impact on the nitrogen and carbon metabolism of the plant, which lead to the change of the grain, the composition. And this is something we report. This one is again like a very long time back. I think this is the first report on the grain quality and the climate change. And so what we said is this is again a small paper in a cereal science. And what we said is the future, the grain quality going to be low in the under CO2 environment. And what we see now is very established. Nearly the world population, if you take 60% of the world population, 6 billion there is some kind of a deficiency of a nutrient, and especially iron, zinc, and iodine, vitamin, etc. And among them, zinc and iron deficiency is a very big problem. It is not only here, it is around the globe. And especially this part of the world, in the tropical and subtropical region, especially developing countries, this is one of the biggest problem. And this also linked to many metabolic diseases, around the health sector. And I will take the protein story. The protein is a very important essential for growth and the maintenance of tissues, and is very important for enzyme synthesis and hormone regulations, and also immune functions. And the protein deficiency is, as I said, is one of the biggest problem. It's nearly 1 billion people is worldwide suffering from a protein deficiency. And what I pick up here is a wheat, because I was working in Australia. I was working with the rice and wheat, the two major crops, but I had to put a little bit of a priority in the wheat, because there was the only funding for the wheat is very dominant. And two major proteins in the wheat, three things: albumin, globulin, and gluten protein. And the gluten protein, especially gliadin and glutenin, are the very dominant. These two determine the bread quality and the nutritional quality as well. And what we did was, we also look at the meta-analysis using all the published data of what we see at the elevated CO2 conditions. In the future CO2 conditions, we could see large reduction in the protein in the wheat and the rice systems. And this figure you can see how in rice how changes take place in the wheat under different nitrogen background. And what we published very recently in Science Advance that in the rice, using all the lot of varieties represented from a Chinese origin and also Japanese, Australian, Sri Lankan, and the global collection, we put into test, we grow them under phase condition. What we see is the reduction of the protein, the reduction is much greater, and also there is a large genetic variability. And what is when we look at the whole proteome, the proteome is also altered. And this is again for the simplicity. We have done a lot of work on the proteome, but I want to share with you, and the whole proteome was altered, and specifically I pick up gluten protein, and gluten subunits. It's a 3 major subunits. Is a high molecular weight gluten is a substantially decreased. And when come to the bread quality, this is the bread we made from the flour. Plants we grow and under elevated CO2, and the same the one of the Australian or wheat cultivar, but we made the bread from those wheat flour. You can see my right side is a small bread is coming from the wheat grown and the future wheat, under elevated CO2. There is a big impact on the wheat bread quality. This is not for us. In a lot of nearly more than half of the world population is a wheat eaters, and the bread is the main food they use, and the quality is a significantly changed. And then the challenge, I have a very limited time, I'm just quickly wrap up, and the protein, what we look at, can we address this issue to how to tackle this the protein concentration in the grain. So then we came up with we know the nitrogen assimilation biology of the wheat plant and the rice plant as well. So we have seen there is a rhythmic assimilation pattern, ammonia and nitrate, sometimes they switch on, they go together, and we design the fertilizer and with the nitrate and ammonia, and when we get those the combined together, so we were able to overcome this issue, like the reduction in the protein concentration. We could get back the protein into the right level. So after that, I had the opportunity to develop a nano nitrogen fertilizer based on the same concept. So now we have developed a nano nitrogen hybrid nitrogen fertilizer. So this is in a process of now patenting and commercializing, and which is a slow release. This is a kind of a dynamic release pattern, and we can manage the whole nitrogen business in one application. So this is what we are heading especially in a mitigation and also adapting to the nitrogen issue of the driven by the climate change. Quickly, I will go into the zinc problem. The zinc is again the biggest global issue. Is zinc micronutrition. It's not only Sri Lanka. It's a global issue, and what we see is zinc used for the DNA synthesis and also is a growth, hormonal biosynthesis, and many other involved with many metabolic reactions, and also so part of the like a large number of enzymes. So deficiency of symptoms, the deficiency of zinc lead to many metabolic related diseases. So therefore, it's a high time to address and tackle and look at the impact. And there is 3 major factors contribute for the low zinc in the cereals and pulses. One is a low soil zinc concentration, and plant cannot absorb. There is not enough zinc in the soil, and also the plant breeding. And over the years, after the Green Revolution, the plant breeding focus was on the yield rather than quality. So this is one factor, because of carbohydrates, we loaded a lot of carbohydrate into grain, and then there was a less attention for micronutrient. And then the third one is the climate change. We have established CO2 and climate change has a major impact. I quickly share this one. This is last 1800 nearly, the rice seeds they're generated in one of the oldest experimental station in Rotham State in UK. You can see grain yield. There was consistent up to the Green Revolution. There is no change very much, but after there was a big change in the grain yield potential. And then you can see zinc and copper and manganese and iron all start to decrease. This is a very classic example, the breeding. The breeding is for the yield, we have really compromised this thing. And so this is the first report again, like we published in around 1995. So we first said that there is issue, the climate change driven issue. There is a translocation issue. The zinc, especially move into the grain, is limited under elevated CO2, and there is a reasonable amount of zinc sitting in the shoot and also leaf blade, but there is a issue that cannot get back into the grain under future climate conditions. And what it is, like we then in 2014 we published this, all the work we published, collected all the data, is largely our data coming from our own experiments, and put together and published in a Nature that we reported that climate change has a major impact on the micronutrient nutrition and the protein nutrition. And this is the meta-analysis we did, and it's a whole lot of coming from our own data, and we can see how diverse the response, and it's a species dependent, and so there is a big issue that need to be addressed. And again, same story with the rice. Rice also substantially decreased this nutrient, but luckily there is a genetic variability. And this is again like we put together this, we want to look at the magnitude of this problem. And as Professor Achini here, like she was the visiting fellow in my lab in Melbourne, and so we put together, we assembled, we got all the published data, and we look at how the impact, how geographically distributed this impact, and especially what kind of the factor contribute for the reduction in this micronutrient nutrition being looked at, and this is in a process of taking into the Nature Food, and we are working on this paper. And quickly, all the overcome to this issue is one of the biggest challenge, and the biofortification is suggested to be one of the strategy. And we can go for a number of pathways. We started more physical fortification. It means that enriching the micronutrient concentration in the grain, and agronomic mean like we add as a fertilizer. And the second pathway is go through the genetic fortification. We can improve the genetic potential and get through genetics mean into the grain. And other thing is a physical fortification. Is my research programs is going for three of the pathways, but the to take into the rest of the world is a genetic pathway is much easy, because we need to establish mechanism, and once the mechanism is established, we can really quickly correct this issue quickly, and we can adapt as well. So we establish the whole transport mechanism of the uptake or remobilization and into grain loading. You can clearly see there is again large genetic variability in a zinc distribution in the grain. You can see a lot of zinc sitting in the allele outside of the grain allele, and lot sitting on the embryonic region. And this is a classic work we very recently did in Australian synchrotron, using what we look at the grain, and we look at where the major bottleneck into the get the zinc into the inner endosperm. And in parallel we have another genetic experiments and many other experiments. And what we see is we identify crease area is a biggest factor, the size of the trees, especially in the grain. There is a special loop that nutrient transport into the grain, and this kind of I think the first picture in the literature in this detail. And what we see is in the size of the trees also very important, and the thickness of the trees is very important, and you can see the varieties that can accumulate more zinc in the grain, they have a bigger tree size, and also their genes, specifically the YSL family gene in the crease area is expressed well, and now we are looking at to get this corrected through using a CRISPR-Cas9, and we using the range of technology and correct this issue and develop wheat varieties and rice varieties which have a higher zinc concentration in the future, and that there is a lot of opportunity to collaborate, and my Chinese counterparts and Australian team also very interested, and we are discussing to establish this work. And final message is for all that Sri Lanka is a natural laboratory. I invite all my international colleagues to come and work with us, and I haven't seen anywhere else in the world this diversity. And we have more than, our land mass is somewhere around 65,000 square kilometers, and we have over 50,000 microclimate systems. Each one is different. And the biodiversity is the highest on the world. It's recognized as a biodiverse hotspot, and and on top of that, the population is 50% directly or indirectly around agriculture. So there is plenty of, this is a laboratory for me. I think for everybody there is plenty of opportunity to do research. And unfortunately, for last maybe 10-15 years, our breeding programs, our average yield potential is not going anywhere now. It's a kind of stagnated, and I think we have pushed the best, and it's you can see the rice yield is like 2010 4.5, like even 2018 4.5, even now is 4.5, 4 to 4.5, it's going around. So that increment is not around. And same story with the maize and other crops as well. Other crops, when you see, we can see that our yield potential is much lower. Rice is at least in that bracket. So I think we need to address this issue. If we want to address the food security and nutrient security, we need to address this issue. It's biologically, I take a China example. Chinese like a hybrid rice. The average yield potential is somewhere around 10 tons per hectare. And so they are pushing to 18 tons per hectare. And like other part of Australia, some places they get 18 tons, 15 to 18 tons. Now it's not, it's always the same, but this biological potential is there. It is, I think, is a high time to push, to little bit in this biological potential, this opportunity there. And So, for me, this is if we increase, our average is 4 tons per hectare. If we increase 1 ton per hectare, so we have somewhere total, Yalla Maha, we call the 2 season, we have nearly 1.326 Ks of the hectares, is 123,000 of hectares. So if we increase the yield potential by 1 ton, the return will be nearly $650 million in 1 year. This is a big income for the poor farming community. Actually, if we invest this money for this program, we can the return is within the same year you can get that type of return. I just want to put it in a simple way. There is an opportunity, to put the genetic potential, and so what we miss is the green revolution is like, okay, we got, and there was ups and downs. But again, the second green I call second generation of breeding technology, mark-assisted breeding, we have not captured fully. And the third generation genomic science selections, I don't think we have captured fully. And the gene editing, CRISPR, Cas9, the genome editing technology, we have not captured fully. And now we are in the era of the synthetic biology and advanced gene editing, etc. So we have not taken advantages of this technology. That's why we lock in that 4 tons per hectare place. So we need to help to push that genetic potential in our agriculture system. At the same time, I call the E and G interaction is a driving force for the agriculture and sustainability, food security. And the E also we have not fully capitalized, because we need the technology. Now precision agriculture is all over the world is used, and the soil management, we have to manage our soil. Most of the soil being degraded, and same time the crop protection, integrated crop protection strategies we need to adapt. And nutrient management, it means that high-end third, fourth generation fertilizer need to come into the system, and the robotics and the post-harvest technology, artificial intelligence, all these things are used in agriculture that need to be capitalized. I'm not telling that is not used, but we need to use extensively. And what I suggest is that it's a high time to push our biological potential, and equally high time to put a big effort to optimize our agriculture, to put this thing together and push the food security and nutrient security, not only this country, in the world. So thank you very much. This is the work done is all over the Australia, Japan, US, and many collaborators in all. I don't want to go into the detail and the list here. And again, thank you very much. I really hope you enjoy the conference.